In this module, on the applications of gene technology, we shall be looking at the principles of genetic engineering, the polymerase chain reaction, genetically engineered microorganisms, genetic markers, antibiotic resistance genes and the use of replica plating, large-scale culturing of genetically modified bacteria to produce useful substances, use of genetically modified animals, and gene therapy. In the 1950s, Crick and Watson, using Rosalind Franklin's X-ray crystallography results, worked out the basic structure of DNA and put forward a model for how DNA replicates and how genetic information may pass out of the nucleus in the form of RNA. During the 1960s, other scientists provided evidence for these theories and worked out the genetic code. Reverse transcriptase enzymes were found in some viruses, and this meant that RNA could be copied into DNA. Restriction endonuclease enzymes were discovered in bacteria, and the knowledge of DNA that we have enables us to change the DNA in a cell and so change the proteins which that cell synthesizes. This is called gene technology, sometimes called genetic engineering. In principle, genes are taken from one organism and inserted into another. There are many ways of doing this, and there are many applications for health and food production. Let's use insulin production as an example. One form of diabetes mellitus, also known as sugar diabetes, is caused by the inability of the pancreas to produce insulin. This is insulin-dependent diabetes, and people who suffer from this form of diabetes need regular injections of insulin after they have eaten a meal. Until fairly recently, the only source of insulin for such injections was from the pancreases of pigs or cattle slaughtered for meat. This could not produce enough insulin to treat all people with diabetes, and the insulin is not identical to human insulin, so can have side effects. There were also ethical issues, as vegetarians may not have liked the idea of using insulin obtained in this way. In the 1970s, biotechnology companies began working on the idea of inserting the gene for human insulin into bacteria and getting the bacteria to make human insulin. Several different approaches were used, and the manufacture began in the early 1980s. There are many ways that a gene for a specific protein could be obtained. Bacteria have restriction endonuclease enzymes, which they use to cut up viral DNA when they are under attack by a bacteriophage virus. These enzymes are used as each one cuts at a specific sequence in the DNA molecule. The location at which they cut usually involves a sequence of 4 to 6 base pairs and is a palindrome. The sequence of base pairs reads the same forwards on one DNA strand as backwards on the other. EcoR1 is a restriction enzyme found in the bacterium E. coli, and it cuts DNA as shown. This produces a staggered cut and unpaired exposed nucleotide bases, or sticky ends. A restriction enzyme could be used to cut out the length of DNA that is the required gene from a whole DNA molecule, but this is difficult. Insulin is a polypeptide. It is a relatively small molecule with only about 16 amino acids. The sequence of amino acids has been worked out. So, knowing that and knowing the genetic code, an automated polynucleotide synthesizer could be used to make a piece of DNA that codes for the polypeptide. Another approach is to look at the beta cells of the human pancreas. These cells produce insulin, and so there will be a lot of messenger RNA, or mRNA, carrying the genetic code for insulin in those cells. If the mRNA for insulin is extracted and treated with reverse transcriptase, then complementary DNA will be produced. If this is treated with DNA polymerase, more of the double-stranded DNA can be made. Extra guanine nucleotides can be added to the ends of these DNA molecules. Now, plasmids are obtained from bacteria. These small circles of bacterial DNA are cut using a restriction enzyme, this time one that does not make a staggered cut. Extra cytosine nucleotides are added to the cut ends of the plasmid. The insulin DNA with GGG sticky ends and the cut plasmids with CCC sticky ends are mixed together in the presence of a ligase enzyme. The exposed C and G bases join by hydrogen bonds, and the ligase joins adjacent sugar and phosphate molecules of the backbones. This produces recombinant DNA, a bacterial plasmid with a human gene inserted. This plasmid is a vector. It'll carry the human gene into the bacterium. The recombinant plasmids are mixed with bacteria in the presence of calcium chloride, which makes the bacterial surface more porous. The bacteria are also given heat shock treatment, a short period on ice followed by a short period at 40 degrees C, and this helps them take up the genetically engineered plasmids. We need to know that the bacteria we are going to culture to produce the insulin have actually taken up the plasmids with the insulin gene. To do this, we use genetic markers. The bacterial plasmid is not just cut anywhere. Plasmids contain antibiotic-resistant genes. If we use plasmids that have resistant genes for ampicillin and tetracycline, we can, because we know the sequence of these genes, use a specific restriction enzyme and cut the plasmid in the middle of the tetracycline-resistant gene. This means that the gene for human insulin will be inserted into the middle of the tetracycline-resistant gene, and so this gene will not function as it cannot code for the protein, an enzyme which breaks down the antibiotic. A sterile velvet pad is pressed onto the ampicillin agar plate and, keeping the orientation the same, pressed onto another agar plate that contains tetracycline. This second plate is then cultured and we compare the two. The colony of bacteria present on the ampicillin plate but absent from the tetracycline plate is the colony that can be removed and cultured. This colony of bacteria consists of individual bacteria that all have the insulin gene. Thus, the gene for human insulin has been cloned. Many other genes are cloned in this way, and we can build up a library of such genes that we wish to research. These bacteria are then taken and cultured on a large scale to produce insulin. This insulin is extracted and then purified for use. So we have covered the principles of genetic engineering, the use of various enzymes such as restriction endonucleases, reverse transcriptase, and ligase, the use of plasmids as vectors to genetically modify bacteria, the use of genetic markers, antibiotic-resistant genes, and the large-scale culturing of such genetically modified organisms, GMOs, to produce something useful. We have also mentioned the role of DNA polymerase. Let's just look a bit more at the polymerase chain reaction. You remember how DNA replicates and that the enzyme DNA polymerase plays a part, making sure that there are no copying errors. The polymerase chain reaction, PCR, makes use of the ability of the DNA molecule to self-replicate and also uses DNA polymerase enzyme. A small amount of DNA, for instance that extracted for a forensic analysis or for ancestry tests using mitochondrial DNA, is amplified. If subjected to heat, the hydrogen bonds between the nucleotide bases will break. If more nucleotides are added, plus the enzyme DNA polymerase, and a lower temperature of around 37 degrees Celsius, then the DNA will make copies of itself. If the cycle is repeated many times, then, from a small amount of DNA, many copies can be made. This technique can also be used in gene screening. For example, in prenatal testing, or in pre-implantation testing for embryos produced by in vitro fertilization, IVF. One cell can be removed from the embryo when it is at the 8-cell stage, the DNA extracted and multiplied using the PCR, and then separated using electrophoresis. A radioactive gene probe is added. This contains a sequence of DNA complementary to a known mutation of the gene. For example, the CFTR gene involved in cystic fibrosis. If the probe sticks, it can be identified by its radioactivity, and we know that the DNA contains an allele for cystic fibrosis. Gene screening can also be carried out on symptomless individuals to find out if they are carriers for certain genetic diseases. Gene therapy is not the same as genetic engineering. Gene therapy can be used to treat, but not to cure, some genetic disorders that show a recessive pattern of inheritance. The individual treated needs to have continuous treatment, and their children can still inherit the genetic disease from them. It can be used for a recessive condition such as cystic fibrosis, but is not suitable for treating dominant conditions such as Huntington's disease, as the functioning allele that is given during the treatment does not replace the defective allele. The defective allele is still present in all cells, and in a dominant condition, only one defective allele of a pair of alleles found on homologous chromosomes is needed to produce the disease. In Huntington's disease, the abnormal version of the protein, huntingtin, causes the neurological disorders that give rise to the symptoms of the disease. Where the condition is recessive, such as cystic fibrosis, then the allele is not producing a functioning protein. Gene therapy can insert the functioning allele into some of the host cells and give some measure of relief from the symptoms. Cystic fibrosis is the most common genetic disorder amongst white Europeans, with about 1 in 20 being carriers and 1 in 2,500 births per year affected. It is possible that being a carrier gives some protection from TB, so that in the past, when TB was rife, carriers would have had a selective advantage. This may be why the incidence of carriers in the population is so high. The gene in question is the CFTR gene, the gene for the cystic fibrosis transmembrane regulatory protein. This protein is a chloride ion channel protein and should be present in epithelial cells of the respiratory tract, the gut, and other tubules such as sperm ducts and oviducts. If the protein is absent or not of the correct shape, so that it is not functioning correctly, then chloride ions cannot pass from these cells into the mucus being produced by the goblet cells. This means that sodium ions will not follow, and so water will also not follow by osmosis. The mucus in these areas is therefore thick and sticky. In the respiratory tract, this means that the mucus is difficult for cilia to waft up the bronchi and the trachea. Bacteria trapped in the mucus can remain and multiply, causing infections. So sufferers need to take many antibiotics. Until about 30 years ago, very few people with cystic fibrosis lived beyond the age of 5 years. Today, with treatments such as antibiotics and physiotherapy and heart-lung transplants for those who have developed emphysema, the prognosis is better. However, adult males with cystic fibrosis may be sterile, as the reproductive tracts have sticky mucus. Gene therapy can also improve the prognosis. Copies of normal CFTR alleles are obtained and cloned. They are encased in small pieces of phospholipid bilayer. The resulting packages are called liposomes and may be used in an aerosol spray or inhaler to place the genes into the respiratory tract of the sufferer. Some of the liposomes will diffuse across the membranes of epithelial cells of the respiratory tract and through the nuclear envelope. They can now be transcribed and translated, and normal CFTR proteins can be made in these cells. The treatment has to be repeated every 10 days, as the epithelial cells are continually being replaced. Other methods of delivering the functioning genes have been tried. such as using adenoviruses that have been made harmless. But there have been some cases of harm caused to the recipients using this method. Although the viruses have been rendered harmless to most, they are being introduced into people who have damaged lungs due to cystic fibrosis, and there are harmful side effects. Of course, there are ethical as well as social and economic issues with the use of genetic engineering in medicine and food production. You will probably have discussed these in your lessons, and you may be required to discuss some of them in an exam. Remember to give pros and cons, and not an emotional outburst. True, we are exploiting animals when we use transgenic sheep to produce medicines, but we have exploited animals for food, fur, skins, bones, and as beasts of burden for thousands of years. Remember also, it is not a valid argument to say that something is unnatural and therefore wrong or harmful. Volcanic eruptions, disease, earthquakes, and tsunami are all natural phenomena, but few, if any, would argue that they are good for us. There is a genetically engineered variety of rice that contains vitamin A and will prevent many children in the developing world from becoming blind due to a vitamin A deficiency. Okay, so now it's quiz time.